Thanks for the introduction, Ken. Today I want to talk about mosquito-borne viruses predominantly. For the last 30 years, since I started my PhD as a QAMR scholar at James Cook in Townsville, I've been working with mosquito-borne viruses, ranging from discovering new viruses or discovering new emerging strains of existing viruses, characterising them genetically or antigenically, elucidating their ecology and mechanisms of pathogenesis in some case. And a lot of these studies have allowed us and given us clues to develop new diagnostics for these viruses and also to develop vaccines. And I want to talk a little bit about our work with applying existing vaccines to arboviruses that affect humans and animals in Australia, and also developing some new vaccines for some of these viruses. This is a typical transmission cycle for arboviruses. Arthropods like mosquitoes and ticks are common vectors, as well as biting midges like culicordes. And it can be a cycle between the arthropod vector and humans, as is the case with dengue, yellow fever, chikungunya. But you also see many arboviruses use animals predominantly, so they're really zoonoses. And humans infection is a result of a spillover from that transmission between, say, mosquitoes and birds, or mosquitoes and other animals. As well as humans, a lot of these arboviruses cause disease in horses. And I'm going to talk a little bit about a couple of viruses such as Ross River and West Nile in particular that can cause significant disease in these animals. There are a number of viruses that are endemic to Australia. These include Ross River and Barmah Forest, and Andreas Subia mentioned these in his talk on chikungunya virus. And indeed, they come from the same genus, the alphavirus genus, as chikungunya, and they cause a very similar disease. In fact, you couldn't differentiate the disease that Ross River and Barmah Forest cause in humans from chikungunya. You need to have a laboratory diagnosis. We have two flaviviruses also, Murray Valley encephalitis virus, and as Alex spoke yesterday, the Kunjun strain of West Nile virus. These viruses have been shown to produce potentially lethal encephalitis in horses and or humans, but the numbers of infections are much lower. Generally, we see only a few cases in humans per year, as opposed to Ross River and Barmah Forest, where we see up to 10,000 cases combined between these two viruses. We also have a number of viruses that are introduced, and we usually get annual outbreaks of dengue in the northern part of Queensland, where Aedes aegypti is quite common. But these outbreaks are always introduced. There's always a traveller coming into Australia that has the virus, and that sets up a transmission cycle, usually at places like Cairns, where you get a lot of tourists coming in. Japanese encephalitis, we occasionally see introductions, particularly in the islands of the Torres Strait. We believe that that's generally due to wind-borne infected mosquitoes coming in from the western province of PNG, which is not far away from those islands. So it's not an endemic virus. Chikungunya virus, Andreas gave us a very good talk on that yesterday. The cases we see in Australia are always tourists returning from a destination or tourists from an endemic area coming to Australia. So they've been infected outside Australia. We haven't seen any evidence of transmission of chikungunya. However, we do have the vectors. We have Aedes aegypti, we have albopictus that's coming into the Torres Strait, and both these mosquitoes could transmit chikungunya should it decide to come in. There aren't many existing approved vaccines for arbovirus for humans. The yellow fever vaccine's been around for many decades. It's very effective. It's a live attenuated vaccine, and you get one dose and you get annual boosts every 5 years. The Japanese encephalitis inactivated vaccine that Heli spoke about yesterday in the trials in horses, that's also been around for a long time. It's been improved from the mouse brain version into a very pure cell culture derived vaccine, and there'll be a presentation in the posters later on today on that. There is also a live version of the Japanese encephalitis vaccine that's licensed for use in China, and a few other countries, but not in most countries. And that's a single dose, and that can be done with boosters. And in Europe, there's also a tick-borne encephalitis vaccine that's available for areas where that particular disease is prevalent. But I want to talk about these Australian viruses. Ross River virus in particular causes on average about 5,000 cases per year, most of them in this part of the world. Indeed, this year we've seen more notifications than any other year on record, and most of them in this part of the world. But for those of you who are visiting this country for the first time and are going to be touring around southeast Queensland, it's a good time to do it because these mosquitoes are fairly inactive at this time of the year. It's the summertime when you have problems. As Andreas spoke about chikungunya disease yesterday, these two viruses, Ross River and Barmah Forest, produce a very similar syndrome. It's a polyarthritis in the small joints, and you can get prolonged arthritis, particularly in the joints, that's associated with persistence of the virus or viral products and inflammation. Other symptoms include fever, and in some cases a very prominent rash. The interesting thing about Ross River is that unlike the infection and disease seen in infants with chikungunya, there's a lot of the child infections with Ross River are very mild or asymptomatic. So indeed, if you get infected with Ross River as a child, you're probably immune for life and you probably don't develop any disease. And if you're like me, who grew up near the coast of Queensland, playing near salt marshes where these mosquitoes breed, I probably was naturally vaccinated many years ago. There's no approved vaccine yet for Ross River. The treatment really is treating the symptoms. Similar to the treatment for chikungunya, analgesics like paracetamol can be effective in reducing pain. Non-steroidal anti-inflammatories will reduce that inflammation of the joints. But the other thing that's very important is rest. And particularly if you're a person who does a lot of hard physical labour in your job and you like a few drinks after work, that's probably the worst thing you can do if you have contracted Ross River virus. So the advice is to take rest and cut down on the booze, and that will help. There are a lot of sportsmen who have contracted Ross River and had to stop playing sport for 12 months because of the debilitating nature of this disease. Currently, the best prevention is to avoid being bitten by mosquitoes. If you have a house that's near areas where these mosquitoes breed, you screen your house. If you going out at a time in a place where there's likely to be mosquitoes, you protect yourself with repellents and personal and clothing that stops mosquitoes biting you. And the councils in Queensland particularly are very active in spraying and controlling the larvae breeding and the adults breeding. That's a very important part of vector control. As I said, there's no currently available vaccine for Ross River. One of the reasons that Ross River is very predominant, it's found in all states of Australia, is that there's several mosquitoes that transmit the virus in inland areas where there are freshwater habitats. Culex annulirostris is found all over the place and it's a very good transmitter of Ross River. In areas near the coast, where mosquitoes breed in brackish water, it's saltish marsh near the coast, these mosquitoes provide a very good transmission. This one in the northern part of the country and Aedes camptorhynchus in the southern part. So the virus is well transmitted by a number of species. In terms of transmission, Ross River is transmitted not only through a cycle between humans and mosquitoes, but also between a number of animals. So it is a true zoonosis. And probably the most important are marsupials such as kangaroos and wallabies. And we've also shown evidence that brush-tailed possums can have reasonably high titers of virus in the blood, therefore can infect mosquitoes quite effectively. And if you've been walking around the suburbs of Brisbane, you will see these animals very predominantly sitting on people's verandas, in trees, walking along power lines, and sometimes living in people's roofs. So these would be very effective transmitters in an urban area. And the kangaroos and macropods very effective in a rural area. Now, Ross River also infects and causes disease in horses. And this is some work that Helly and Anita Barton have done together. And based on 4 case studies of horses that were infected with Ross River and showed specific disease, particularly disease of the joints, swelling, affecting the lower limbs, and generalised musculoskeletal stiffness. One of the important signs that you see is that a horse that is working or is a high performance horse, when they're infected and have this disease, they're very, very reluctant to train or work. So you have this exercise intolerance, and that's something that's very specific, and a lot of vets have documented this with horses infected with Ross River. If you look at the number of horse samples that were submitted to the Victorian Arbovirus Disease Program for testing for Ross River, there's a high percentage of these horses came back positive. They had something wrong with them and they were shown to be infected with Ross River. And there was a particular spike during the outbreak that Helly spoke about yesterday of arboviral disease in southeastern Australia, with up to nearly 1,000 animals affected in some way, and over 30% of these had infection with Ross River. And if you look at a subset of those horses that were infected during that outbreak, you'll see that a number of states showed a lot of infection with either Ross River virus or other viruses, particularly flaviviruses. New South Wales had predominantly the Kunjun strain of West Nile causing infection, while in Victoria you saw a large number of horses infected with Ross River. So a lot of the disease in Victoria was caused by infection with Ross River. Similarly in South Australia, a significant number. A lot of these horses in yellow were probably also infected with one of these viruses. A subsequent study by Helly and us on a number of samples that were opportunistically obtained from racehorses over a few years. A lot of those also tested positive with Ross River, and some of them have quite high titers of neutralising antibody to Ross River, indicating the significant level of infection in these high-performance horses. Now, because these samples were opportunistically taken, there was no, we weren't able to correlate this infection with any disease. Now, as I said, there's no approved vaccine for Ross River, but John Askoff at QUT, who's been working on Ross River for over 35 years now, has joined together with bats that have produced a formalin and UV inactivated version of Ross River. This is grown in vero cells and 2 doses, alum adjuvanted, and they showed protection in mice. They used both wild-type mice and interferon type 1 knockout mice to show reduction of viremia and protection of lethal effects. They also showed partial protection against chikungunya, which is interesting because these viruses are antigenic related. This virus has actually gone through phase 3 trials, and in approximately 2,000 human volunteers, they showed 90% seroconversion and induction of neutralising antibodies in young adults. If you looked at a small cohort of older people, the vaccine wasn't as effective. We've actually thought about in preparing a vaccine for horses, and we could either go down the inactivated track that John has with the human vaccine, but also we looked at joining up with a collaborator, Dr. Gorman Pilsman from the Netherlands. And Andrea spoke about the baculovirus vectored VLP vaccine that was developed for chikungunya virus. Very effective, and VLPs are antigenically very similar to the native virus. And Gorban's agreed, and he was very keen to make a Ross River version of this. So he's gone ahead to make a Ross River version of this virus so we can actually do some trials in horses. We just need to get the funding to do the assessment in the horses once this vaccine is produced. But this is a very exciting avenue. I also spoke to Mohammed about the possibility of doing some gamma radiation inactivation of Ross River, and that's another possibility. So in summary, just looking at the Ross River section, debilitating arthritis disease affects thousands of people every year in Australia, and there's also documented evidence that it produces a serious illness in horses, particularly making them unable to train or perform. But we need more studies, and we need more studies to look at that and have a clear clinical picture of what Ross River does in horses. There is a killed vaccine available in human trials, and that's one way to go for humans or horses. But the problem is that because Ross River does not cause fatalities, it's restricted to Australia. That vaccine is unlikely to go ahead and be rolled out. Unless Ross River escapes from Australia and gets into the US and causes problems there, we're unlikely to see it. But this vaccine is ready to go. The VLP approach for horses is another avenue that we're looking at. Okay, West Nile virus in Australia. Helly mentioned the outbreak in 2011 of a Kunjun strain of West Nile. The symptoms that were seen were very similar to the symptoms that were seen in the horses infected with West Nile in the US. We had approximately 1,000 cases, probably more than 1,000 cases in all if you look at the unreported ones, and about 15% mortality. This is the distribution of cases. You can see most of them occurred in New South Wales, Victoria, and some in South Australia, so particularly focused on the southeastern part of the country. And this occurred after particularly heavy rains that occurred through the northern, central, and particularly in the southern area. So this is typical response of vectors breeding after these rains, and possibly vertebrate hosts also breeding up, providing that transmission. West Nile virus has a transmission that predominantly between mosquitoes and various bird species, and you get a spillover effect of infection, but these humans and horses tend not to participate in the transmission cycle, but they do develop disease. If you look at the US, we've had over 40,000 cases reported since the virus first was introduced there in 1999. About 1,700 deaths. Horses, there was a large number of horses initially infected, then they introduced a vaccine, and those numbers sort of dropped down. But initially, there was a very high mortality rate. I mean, that includes horses being euthanized, but 30% mortality rate was a very high number, and that's why that equine vaccine was introduced. The virulent strains of West Nile in the US also had a very strong effect on some species, and often people would walk out and find dead crows on their lawn, and they'd know that West Nile had come into that particular area. Unlike that, the outbreak in Australia, as Helly said yesterday, we saw no evidence of bird death, and we saw no human cases of neurological disease associated with infection with this virus. So this was a very horse-specific outbreak. And when we looked at the virus, we did in fact show genetically it was Kunjun virus. It was not an introduction from somewhere else around the world, but it was particularly virulent. Not as virulent as the US virus that can kill mice very quickly. These are 4-week-old mice. And if you look at the normal Kunjun strains, they're pretty transient. They're very attenuated in this mouse model. But this new virus, 2011, was approaching the virulence of the US virus, but not as virulent. That may explain why we didn't see bird deaths in avian and human disease. There are vaccines approved for horses for use in the US and Europe. One of these is a killed vaccine made by Pfizer. I think it's now marketed under Zoetis. A couple of doses of this formalin inactivated product with annual boosters seems to be protective to horses. This virus is now pending approval in Australia to use against Kunjun. Another vaccine that's vectored as a canary pox vaccine produced by Muriel, and this is a pox virus that can grow in avian cells, but it doesn't replicate in mammals. It will infect the cells, it will express the antigens of the virus, in this case West Nile, but it doesn't make new virus particles, so it's quite safe. It also induces a good response, including antibodies and T cells. Again, this is another one that's pending approval. If you look at the comparisons done on these two vaccines in the US, both of them after 2 inoculations produced similar levels of neutralising antibodies, so they're both quite effective with this 2-dose regime. Now these neutralising responses were tested against the US virulent West Nile strains, but when we got some of these samples from this killed vaccine, we showed that they also produced robust neutralising responses to the equine virulent West Nile virus that was isolated in Australia that caused the 2011 outbreak. So effectively these vaccines should work against Kunjun. Interestingly, the killed vaccine, the serum that we got from horses vaccinated with this vaccine, did not neutralise MBE at all. We saw no neutralising responses to MBE, so this may not be effective for cross-neutralisation. Just want to quickly talk about some recombinant vaccines that were developed by Alex. You heard about Alex's Kunjun vectored Ebola vaccine yesterday, but this is a variation on the theme. He developed a DNA vaccine that expressed the PRME of Kunjun virus and showed that that could protect, but you needed a relatively high dose of the DNA to inject into animals. He also developed a DNA vectored whole virus vaccine. So basically he put the whole genome of Kunjun virus in a plasmid and it was transcribed from a CMV promoter. So when that plasmid goes into the cell, RNA polymerase 2 transcribes the whole viral genome, which goes into the cytoplasm and sets up infection. So it's a DNA-delivered infection of a live virus. We also introduced, as well as Kunjun being naturally attenuated, we introduced other attenuation markers to make it a highly attenuated vaccine. And that worked quite well, and we published that in PNAS. But more closely to what he spoke about with Ebola, he developed what we call a SRIPS vaccine, a single round infectious particle. And that's where he introduced the replicon into a plasmid under the control of the CMV promoter, but he removed the capsid. And that meant that it was a replicon, and then he also produced authentic capsid in the same cell so you could get a single round of infection. I'll just show you this a little bit more in detail. The plasmid that contained the replicon with the capsid removed was introduced into the cell, it went into the nucleus, the RNA was transcribed, and you get this replicon that doesn't have the capsid, so it can't encapsulate itself. It can replicate but can't encapsulate. So in that same plasmid, he produced an authentic version of the capsid under a different promoter. So in that transfected cell, you can get encapsulated of this product here. These single-round infectious particles or pseudo-infectious particles can then infect surrounding cells, and this then can replicate and produce more viral antigen, but it can't spread because there's no capsid here. So a very effective way of getting a limited spread of a vaccine. It produced antibodies to the range of proteins and also produced cytotoxic T cell responses. So this is a very effective vaccine, and you can use very small amounts of DNA because it does that initial round of spread. We tested this in horses. We used a gene gun and also intramuscular inoculation. The gene gun just allowed delivery of the DNA into the skin to target those Langerhans cells. We got very good neutralising responses. You can see when you compare the intramuscular inoculation of DNA, we needed a lot more DNA than we did by using gene gun. We got very small amounts of DNA, and we could produce neutralising responses with one dose, and by the time you got to 2 or 3, those neutralising responses were very high. So this is a very effective way of vaccinating horses against West Nile. I'll quickly go through another virus. Summary there. Equine West Nile. We had a large outbreak in 2011. We had 1,000 cases and significant mortality. There are some West Nile vaccines that are approved for use in the US and Europe, and they would be effective against Kunjun. We have some novel experimental vaccines that are also effective. But these viruses, these outbreaks are unpredictable. We still don't know why this outbreak occurred because that virus has remained around in the same areas, but there's been no outbreak. So there's a combination of ecological, environmental factors there. Quickly run through Murray Valley encephalitis virus. This is a related virus. Again, it shares the same sort of transmission, produces similar sort of disease in horses. Predominantly found up in the north. You get cases, human cases up here, and then occasionally you get other cases, and rarely we see the virus move down into the south, usually after a lot of rainfall. The disease, one thing to note is that there's a very low case to infection ratio. It could be as low as 1 in 1,000 people infected actually get any disease. The disease is a typical encephalitis. A number of things can relate and result in death. One of the factors is that a lot of the people who actually recover have permanent brain damage, and this could be motor neuron damage, it could be mental impairment, or it could even be psychiatric problems. And there's about 10% mortality if treatment like supportive ventilation is available. And there's no vaccines available for this virus. And my colleague Mario Lobigs, who Nick talked about yesterday, and we did some collaborations, and we produced a number of experimental vaccines using recombinant vaccinia viruses, DNA-based viruses, alpha virus vectored viruses, and all producing the PRME, the structural proteins on the surface of the virus that form a subviral particle that's very antigenic. And these all worked extremely well, and you could remove the cytotoxic T cells of the mice after vaccination and showed that this was an antibody-mediated protection. But less than 20 human cases in the last 5 years, really you can't make a vaccine to a virus that's uncommon. But some studies that Mario did, and this is a collaboration with Mario, Nick and I, that we actually did some studies in mice initially, and then we actually used the killed vaccine that was the killed JE vaccine that was adjuvanted with the Delta Inulin, Advax, and showed that we could get good responses in horses to JE after 2 doses, but also we could get robust neutralising responses to MVE. This is the precursor of the study that Helly did in the foals. And we also saw some protection against West Nile. So there was really good evidence that this might be a good vaccine against MVE in horses. That was just prior to that outbreak in 2011, so that result was quite intriguing. Just a summary, MVE affects humans and horses in Australia. There are large outbreaks in southeast Australia. They're rare, and there are too many cases to warrant a vaccine made to MVE, but we do see cross-protection to MVE using a JE vaccine, particularly if it's well adjuvanted. That's the important part of it. So in conclusion, we've got a number of viruses that can affect humans and horses in Australia, and these may be exacerbated in the future, particularly with climate change. If we get more rainfall in certain areas and warmer temperatures, this could be a factor that could kick off these outbreaks more commonly. There are existing vaccines to some of these viruses or related viruses that may provide protection. It's a matter of should these vaccines be approved for use in Australia, either for human or veterinary use, and that will depend on how a lot of factors in the future. Could they be used in combination with other vaccines? And there's a range of vaccines that are currently used, Hendra, tetanus, strangles, and perhaps some of these vaccines could be incorporated in a combination vaccine. Just some of my collaborators, particularly Helly, who was involved in a lot of the work you saw here, and she and Anita for providing some unpublished data on Ross River. My colleague Alex and a number of members of my lab, Chris Pollitt for doing some horse vaccination, and David and Justin who were involved in the thrips vaccine. Mario was involved in a lot of the work that I've shown you here. Peter Kirkland was our collaborator at EMAI in New South Wales, particularly providing a lot of horse samples. Nick was involved in a lot of the vaccine trials using the Advax JE. And Gorban, who's come on board in producing the Ross River VLP vaccine. Thanks. Vaccine produced by Muriel. My understanding that it's a 2-dose regime that produced those responses. So it's the same yes, and it doesn't need an adjuvant because it acts like a bit like a live vaccine. Yeah.
Okay, I think in the interest of time we'll move on. So.